Guys, we're going to take again a picture with the sticker, but in this case, I need to look in the back over there. So, under the picture, please.
Right. Today we are receiving Dr. Williams Beckerson. He's a MSN postdoctoral research fellow trained in molecular biology and genetics. Dr. Beckerson has two major research focuses. The first one is molecular genetics work with the zombie fungus, cordyceps, and zombie ants. And the second one is DBER research working with to identify best practices for implementing active learning in the biology classroom. He obtained his bachelor degree in Georgetown College with his major in science in biology and a minor in business management. Later, he attended the University of Louisville, where he obtained his PhD in philosophy in biology. Dr. Beckerson is an active member of the community and has several media outlets dedicated to helping educate others about various topics in biology, including TikTok, Skype, A Scientist, and even Zombie and our video game. So please receive him with a big applause. So, Dr. The forum is yours.
Thank you very much. It's a pleasure to talk to you today about some really cool zombie science today. I have a little bit of everything in here. So if you're not a biology major, stick around. There's some stuff in here for you. If you are a biology major or somebody well-versed, we're going to cover some topics that you are probably familiar with, but hopefully these topics will cover material in a new light and help you to see them in a way that maybe you haven't learned about before. So I think a good place to start whenever I go out and talk about zombie science is to define what we mean when we use the word zombie. And we do use this term actually in the scientific literature. And so it's a good idea to define what we mean before we get started. Because when you think about the term zombie, you probably fall into one or two categories based on the type of film and pop culture media that you consume. And there really is kind of two camps of thinking here. So the traditional view of a zombie is, of course, simply the dead brought back to life. And there are plenty of great shows and movies that are really fun to watch. I'm a big fan of series like The Walking Dead. But when you think about this definition of zombie from a biological perspective, there's a lot of issues with the real life science that goes into this definition. Just for example, once something has died, deterioration kicks in. So separation of ligaments from bones that tether muscles to the skeletal system that allows you to move your body becomes difficult. So there's a lot of questions about like if you could even reanimate something, how is it even moving around? There's problems with metabolism. So even if this dead creature brought back to life could bite and eat human flesh, how is it digesting this flesh without the proper digestive system? So, when we start to think of things from a science standpoint, these movies are not very biologically relevant. However, there is a sort of subcategory of zombie movies where rather than the dead being brought back to life, what we see defined as a zombie is a living organism that has been infected by a parasite or a pathogen of some sort, usually a virus or a bacteria, or in some cases, fungi. If you're big fans of the new HBO series, The Last of Us, you're going to really enjoy this talk because we're going to talk about some of these behavioral manipulating fungi. But under this classification of zombie, things become a little bit more biologically interesting because these issues that we run into with the dead brought back to life are no longer such a problem. And when you compare and contrast the two, you'll find that there's a lot of similarities. However, a lot of times these types of zombies are even portrayed differently. So a zombie from, the Night of the Living Dead is kind of your classical slow wandering, mindless creature, whereas a zombie from I Am Legend is very fast and aggressive and really exhibits normal behaviors that a human would exhibit, but pumped up to level 11, right? So under this classification of zombie, there are some real world examples. So as it turns out, there are real life examples of pathogens and parasites that manipulate host behavior in this way. Now, I mentioned that most of the time in these movies, viruses are the culprits for these zombie outbreaks. And perhaps a good place to start is to talk about the rabies virus, which you can see here in the bottom middle panel. So this is the rabies virus, and this is a rabid dog infected with said virus. And when the rabies virus infects mammals, it causes a couple things to happen. The first thing that it changes is a physiological response, meaning it changes the physical way in which the organism works, in which it causes the act of swallowing to become incredibly painful. And this is why you often see the foaminess in rabid dogs' mouth, because they're intentionally not swallowing due to the pain that causes them. And this is advantageous for the virus because the virus reproduces and spreads through saliva. So by causing this physiological change, it's actually building and accumulating more virus particles in the mouth. Now, in order to spread, it also increases aggression. So a lot of times you'll see these rabid dogs become very irritated and are more prone to biting behaviors. And this biting behavior helps to spread the saliva to new hosts. So what you're going to see throughout the theme of this entire talk is all of these behavioral manipulations have evolved particularly because they increase the spread of the parasite or the pathogen. So another example of a mammal infecting behavioral manipulating parasite is Toxoplasma gondii, which maybe you're familiar with. And the natural system for this protist is cats and mice. So when it infects mice, it causes behavioral changes that cause them to be more brave, if you want to think of it that way, or more willing to approach the scent of cats. And this is obviously bad for the host in this case because cats eat mice. And this is what the Toxoplasma wants, because the main host in which Toxoplasma reproduces is felines in their digestive tract. So by being picked up in the natural environment by mice and then causing them to be more likely to be eaten by cats, they're increasing their chances of successfully completing a life cycle. Now, there's lots of other examples that I've got here. It's not just pathogens for mammals. And as it turns out, most of the behavioral manipulating pathogens actually infect invertebrates, which are those organisms that are all around us that don't have the spine and the brain column like we do. And a great example of that is the amber snail, which is infected by a parasitic worm. And this parasitic worm, just like the rabies virus, causes physiological and behavioral changes. The behavioral change that it induces is called summit disease. This is going to come up a couple times in this talk because it's a very common behavioral manipulation that we see across many of these different organisms. And what summit disease is basically the desire to want to climb things. So to get really high up in the air. And this is important for the reproduction of this worm because the main host of this worm is actually the digestive tract of birds. So just like the Toxoplasma, this is a dual host system. It infects first primarily the birds. It's then pooped out all over the place, picked up by snails, and the snails can become infected. And then the goal is all about getting back to the bird to spread further and reproduce. So after the summit disease occurs, this particular worm starts to do a little dance. inside the eye tubes that are usually translucent in the snails, and it makes it very visible to predatory birds. So being in that elevated position with this basically large neon sign saying, "Come eat me," all but ensures that this worm will complete its life cycle. Now, I could go on and on about all the really fascinating biology of behavioral manipulation, but for the sake of time, we're going to talk mostly today about my favorite group, which is the behavioral manipulating fungi, that most of which infect insects. Now, to talk about how these fungi actually manipulate their hosts, I want to very briefly go over a basic biological principle that many of you are probably familiar with, and that's symbiosis. So if you're any Marvel fans out there that are super into Spider-Man and you remember the Venom and the Carnage series, and those are called symbiotes. They're called symbiotes because they interact with their host, either Peter Parker, for example, or any of the other hosts of characters. So it's a good way to remember the word symbiosis if you're a nerd like me. But there's basically three flavors. So the first is called mutualism. Now, this is a win-win interaction. So when things are mutualistic, both individuals interacting with each other benefit from that relationship. And pollinators are a fantastic example of mutualism. So in this example, a bee will land on a flower and it will pick up pollen from the flower. And when it goes to another flower from the same species, it will deposit that pollen. And this aids the plants in their reproduction, increasing their reproductive fitness. The bees, on the other hand, do this because they get nectar and sugars and carbohydrates from the flowers. So it's sort of like a trading system. The bee is picking up a meal and taking that back and making honey. And in return, it's helping the flower to reproduce. So both individuals win. Now, the second type is a little bit more rare. It's called commensalism. And you can think of this as an indifference interaction. And there's actually kind of two groups of thought on this, whether or not commensalism actually truly exists in nature. Because when you get down to the really nitty gritty, it's hard to define an interaction where one party is not affected. However, I think a really good example of this could be tree frogs. So tree frogs are very adapted to the environment that they live in, and they have evolved to look very similar to the types of trees they associate with. And trees are living organisms. So these frogs rely on these trees for elevation and camouflage, and so therefore get a benefit of deterrence for predatory animals. In return, the tree doesn't really benefit, but at the same time, the presence of the tree frog also doesn't hurt the tree. So this might be one example of what we would call commensalism. Now, the third type, which is my personal favorite because of my particular line of research, is called parasitism. Now, this is the one-sided interaction, or an interaction where one individual benefits at the expense of another individual. So leeches are a fantastic example of this. Actually, in the English language, leech has become associated with somebody or something that like takes from somebody or something else, because that's exactly what's occurring in parasitism. And leeches are a fantastic example of a parasite. They suck the blood of another organism that hurts their host, but gives them a benefit. Now, you may have heard me use the terms parasite, which basically is an organism that undergoes parasitism, and pathogens sort of interchangeably. So real quick, I want to kind of give you an overview of the similarities between those two things. And you can basically boil it down as not all parasites are pathogens, but all pathogens are parasites. So the term parasite just refers to an organism that undergoes a parasitic relationship with a host, whereas a pathogen in particular can be vaguely described as a microscopic parasite, right? So a microorganism, if you break it down into the Latin roots, is a microscopic, meaning really small, organism, which is a living creature. And there's basically 5 groups of life forms that can fall into this definition of microbe. There's bacteria, which you're all very familiar with, I'm sure. Archaea, which have a lot of similarities with bacteria, but are actually more closely related to eukaryotes, but still they're single cellular, and they often live in really extreme environments. There's fungi, algae, and also protozoans, which are single cellular kind of animal-like cell organisms. Now, in addition to these organisms, we also lump in viruses into this category of pathogens, even though viruses are not what we would define as living organisms. And this is just because, a while back, biologists arbitrarily picked a couple defining factors for what is living and what is not living to separate things like cells from basic things like chemicals. And when you talk about viruses, they don't meet two of these criteria. The first is homeostasis, which refers to the ability of a living organism to maintain its environment. So if you think of things on the macroscopic scale, humans are able to regulate their body temperature. We have blood and lymph and all kinds of fluids that are kept inside our body by our skin and away from the stuff outside our body. So we can perform this kind of homeostasis. When you think of things at the cellular level, it's the same thing. Cells are able to keep certain chemicals outside of the cell membrane and certain things on the inside and maintain that imbalance called homeostasis. Viruses do not do this. Viruses are simply protein capsules filled with DNA or RNA. Also, viruses don't have the capacity to grow. They rely on other living organisms to even do basic functions like reproduce. So while a cell might eat, if you want to think of it that way, obtain nutrients and chemicals and actually grow bigger and then divide to become smaller, and that's how they replicate, viruses don't have the capacity to change in that regard. But anyway, to sum everything up in our quick little introduction lesson, viruses are lumped in together with other microbes and are called pathogens when they have a parasitic relationship or a harmful relationship to a host. Now, we're going to talk about pathogenic and parasitic fungi today. And fungi do all sorts of really cool symbioses. There's mutualistic fungi, like the mycorrhizal fungi that associate with the root base of plants. And actually, just like the bees, form this sort of barter system where they're trading minerals and water, and in exchange, they're kind of eating a little bit of the roots and getting carbohydrates and sugars in return. There's also commensal fungi. If you want to consider lichens a fungi, it's a little bit cheating because they also have some photosynthetic algae lumped in there, but they operate much the same way that tree frogs do. They grow on the trunks. They don't really hurt the tree, but they get an elevated position for more light and thus benefit. And finally, they have tons and tons of pathogenic relationships, particularly with plants. This is an example of a rust that infects cereal crops, so things like grains, and it can cause billions of damage to agricultural profits every single year. So there's a lot of effort to understand how fungal pathogens interact with their hosts and figure out ways in which we can kind of help these sick plants and other things. But fungal pathogens are not just particular to plants. There's plenty of fungal pathogens for animals as well. Oral thrush is an example of usually found in people with suppressed immune systems, where yeast can overgrow in the oral cavity and present itself like this. There's also things like athlete's foot, which grow in damp and warm environments, hence the name athlete's foot, coming from gym socks that are wet and warm. And then there's also really ecologically important fungal pathogens like chytrids that can wipe out entire populations of amphibians in a very short period of time. So there's a lot of impetus to really study how these fungal pathogens interact with their environment. And when we think about fungi in general, there's kind of two different portions of the fungus that make up the life cycle of all fungi. The first is the fruiting body, and this is probably the part you're more familiar with as a mushroom, for example, because you've probably eaten these in your salad or had these on your pizza, right? This is the reproductive part of the fungi. But when it comes to the total biomass, most of the biomass is actually underground in these large web-like structures called mycelium. And it's these mycelium that do all the very interesting chemical things that fungi like to do. So fungi interact with their environment in an external way. If we take digestion, for example, fungi actually do external digestion. They grow on their food and they secrete chemicals outside of these mycelium that break down the food that they're on. And then they absorb that nutrients to continue to grow. This is in contrast to animals, which do something you might consider to be internal digestion, where we put food into our bodies, secrete chemicals to break it down, absorb it, and then excrete it. Instead, fungi live on top of their food. But just like they do external digestion, fungi are also renowned for making what are called secondary metabolites, or chemicals that are not required for survival of the fungus, but play really important roles in their ecological niches. And zombie fungi are just one example of very cool ways in which fungi have evolved to secrete chemicals that manipulate their host for this, in this parasitic example, and actually benefit the fungus in its reproduction. So let's talk a little bit about zombie ants. So zombie ants are not a term used for all ants. In fact, they're mostly made up of carpenter ants. And this has to do with simply the amount of biomass that's available in ants. So carpenter ants compared to other ants are particularly large. All right. So they're hundreds of times bigger than sugar ants. So when you think of the fungus and the goal of the fungus being to produce a fruiting body or a mushroom to reproduce, you need biomaterials to do that. You need energy. So you need a meaty girl to be able to eat and get enough nutrients in order to form a mushroom with spores. So it's no surprise that it's really these larger ants that are affected by this fungus. Now, you might think, well, isn't that kind of restrictive, just infecting, one genus of ants? And I'd say that's a fair question, but here I present to you a map, and everything on this map labeled in green is where you can find ants from the genus Camponotus, which make up the carpenter ant. So as you can see, it's really ubiquitous. The only place you really don't find carpenter ants is on the polar ice caps. So there is no lack of food for this fungus. Interestingly, though, when you overlay a map of where you can find the fungus, you'll see that the fungus is not as ubiquitous as its host. Now, the second map I'm presenting shows all the instances of Ophiocordyceps, which is the fungus that causes zombie ants, that we have found on an app called iNaturalist, which is something that I'll actually present to you towards the end of the talk. But I want to point out that this is by no means a complete map. There's a lot of discovery being done. But just from what we have already accumulated data-wise, you can see that there are particular regions of the world where we don't find zombie ants. And the thing that those all share in common is they are very dry, right? So again, just like athlete's foot, Ophiocordyceps requires not only heat, but also moisture. So you're not going to find them in very dry places like the Midwest or the northern parts of Africa or Eastern Europe or Middle East. And you're also not going to find them in really cold areas like the northern parts of Canada and Russia. Because just like Goldilocks, Ophiocordyceps is a very particular species of fungus. And it has evolved to do some pretty spectacular things, but at a cost of being less adaptable. So let's talk about the life cycle of zombie ants. So, how does this all work? Well, everything with a fungus begins with a spore. So here I have a cross section cut out and shown for you of this fruiting body of Ophiocordyceps. And you can see here the long wispy spore-like structures. Now, these are considered ascomycete fungi. So going back to your biology course, the Latin root words for ascomycete refer to them as sac fungi. And you can see here in this picture, these little sac structures that are housing all of these spores. So there's your hint that these are ascomycetes. Now, a really fascinating thing about the physiology of these fruiting bodies is the spores aren't just released onto the wind. They're actually fired out like harpoons. So they get a little bit of distance just from being ejected from the fruiting body. They can then float on the wind. And because these zombie ants are in a high position, they can float pretty far before either landing on a new host or simply landing on the ground and overwintering in their durable spore form until a new host steps on them or picks one up that way. Once a spore comes into contact with a compatible host, it then grows inside the host and starts to secrete chemicals that change the behavior. Now, there's three really notable behavioral changes that we see in zombie ants. The first is the presence of antisocial behavior, pun entirely intended. So ants themselves are very social. They perform very unique roles inside the nest, so much so that they've even evolved to have different castes, which are physical differences that are associated with the roles that they play. So on the left here, you can see this is a miner girl from the colony, and she can play roles like foraging. She can go out and look for food and bring back water, or also nursing roles, taking care of the brood inside the nest. Now, this girl on the right is called a major, And these girls play defensive roles. They are guardians. They protect the nest from foreign insects and things that would destroy their nest. And they also do some of the heavy lifting as well. Now, what we see is in both cases, if either of these castes are infected by Ophiocordyceps, you see that rather than doing their jobs, they leave the nest and they perform, basically abandon their social roles. Now, this is really important. This is a really important behavioral manipulation that has evolved in the fungi, particularly because of the way that ants deal with sickness in the nest. So ants have a really effective but sort of barbaric approach to illness, where when they smell that another ant is sick, they will kill it and take it really far away from the nest so that it can't spread whatever is infecting it to the rest of the colony. Now, because of this, if you're a zombie fungus and your whole goal is to get the ant to go up high so you can spread your spores far on the wind, being decapitated before you've even had a chance to develop your fruiting body is kind of the end of the road. So it's, of course, obvious that these types of antisocial behaviors would evolve. Now, the second behavioral manipulation that we see is summit disease. So if you remember back to the amber snail and the zombie snails that we talked about, summit disease is a really common phenotype that we see in all types of behavioral manipulating parasites. So just like the amber snail, these ants are made to really prefer climbing and to hang out on the really far peripheries of plants where there's really small twigs. And this ultimately leads to the third and probably the most famous behavioral manipulation in zombie ants, which is called the death grip. So the ants actually bite down onto the substrate that they're on and cannot let go. And they're still alive at this point. We have videos in the lab of them trying to, pull themselves away, but they can no longer release that grip once it's established. It's at this point where the fungus, which up until now has been growing as single-celled yeast-like cells inside the ant, starts to connect and form its mycelium through things called anosmosis tubes. So basically all the little cells form little bridges and connect to one another. And they start to eat and digest the ant from the inside out. And they use all this energy and siphon it all up to that point at the back of the head to grow the fruiting body, which then is populated by spores and repeats the cycle. And here's a clip of that, or a time lapse of that happening from BBC. So you can see the ant has firmly gripped onto a twig at the very end of a tree. And soon after, the ant is killed by killing compounds, and then the fruiting body is grown from the back of the head. So when it comes to behavioral manipulations, those three were pretty well established in nature. But once we brought these organisms into the lab setting and started to take a closer look, we realized that not only are a lot of these behaviors shared among different behavioral manipulating pathogens, but there's also some more subtle behavioral changes that we see as well. The first of which I'm going to show you is up here in the top right. And this has to do with the circadian rhythm of the ants. So on the left, you'll see a container of uninfected ants, and on the right is a container of ants infected with Ophiocordyceps. Now, these ants are nocturnal, so you see when we turn the lights on, the uninfected ants on the left will go into their housing tubes and go rest. But if you take a look at the right side of the infected ants, you'll see that the foraging behavior and the wandering behavior continues even during the daylight when they would normally be resting. So just like the zombies that we see in film and pop culture, these Ophiocordyceps infected ants are continuously wandering regardless of time. Furthermore, also like the zombies that are portrayed in film, we see during late stage infection major issues with motor coordination that occurs in ants. And this has been called the drunkard's walk due to the fact that it looks like the ant is extremely drunk, by Dr. Hughes at Penn State. And this is unique to Ophiocordyceps infections. There are other types of fungal pathogens that are more generalist in nature that when you infect these ants with do not cause either the circadian disruption or this drunkard walk behavior. So these are very particular behavioral manipulations that we're just now trying to understand in a lab setting. So the way in which the fungus is able to cause these behavioral manipulations has a bit to do with the way in which it grows inside the ant. So when this infection takes place, we were curious, like what's happening inside during behavioral manipulation? In particular, Dr. Charissa de Becker was the first to really start to take a deep dive into the molecular work and see what's happening in here. And what she found was inside the ant during infection, the fungus will grow and eat the muscle and the fat tissue, but will completely avoid, and we think may actually in fact preserve, the neurological tissue in order to be able to secrete chemical compounds that change the behavior of the host, which makes a lot of sense. If you immediately start to destroy the neurological tissue of your host, you're not going to be able to cause it to do a lot of things that are beneficial for you in the long run, similar to the way that in which these traditional view of zombies doesn't make a lot of biological sense. And here's an electron microscope image from Dr. Hughes' lab that shows some of these fungal cells now connected by these anosmosis tubes growing into the muscle fibers and consuming the muscle tissue within the zombie ant, but avoiding the neurological tissue. Now, when we talk about zombie ants, everything I've showed you up to this point in terms of research has really just been done on one or two species. But as it turns out, the more we keep looking for these things, the more we realize that there are dozens, if not hundreds, of different carpenter ants that are infected by separate species of Ophiocordyceps. In fact, our collaborator, Dr. João Araju, who works at New York's Botanical Gardens, he's gone to, just over the course of about 5 years, he goes to South America and comes back with 10, 12 new species every time, and then publishes on their differences in morphology and behavioral manipulations and also the host specificity, or which hosts are infected by this fungus. So really, zombie fungi are not one species that infects all of these carpenter ants. They're actually a collective or a genus of many different species that are very host sensitive. So to kind of hit home this point, I want to show you, here's 3 examples of closely related ant species. So they're all carpenter ants. They're all from the Camponotus genus. And here's the 3 associated Ophiocordyceps species that affect or infect and manipulate these ant hosts. Now, what's interesting, in a lab setting, even with lots of controlled variables, if you try to infect the ants from a different species of Ophiocordyceps, you'll see that the Ophiocordyceps simply cannot complete its life cycle and does not cause the behavioral manipulations that are common in its original host. So we call this host specificity. Host specificity is just defining how many hosts you can infect. If you're very host specific, then you, like the Ophiocordyceps are, you might only be able to infect one species, even if it's very similar to other species of ants. If you're more generalist, you might be able to infect lots of different insects. There are pathogenic fungi like Bavaria bassiana, which can infect pretty much all insects. So it's a very generalist fungi. You may also be familiar with the concept of host specificity from coronavirus. So the coronavirus was suspected to originally infect bats, and not just COVID, but also other SARS viruses are known to infect bats. And it is a little bit more general because it was able to jump hosts and infect other types of mammals as well. Now, we think a very similar, we would call this type of shift in hosts a host shift. And we think a very similar thing happened with Ophiocordyceps based on genetic data. We think that Ophiocordyceps first emerged in beetle larvae, which it actually infects underground and then grows up out of the ground, hence the long snake-like structure. And that's actually what lends Ophiocordyceps its name. The Latin roots for Ophiocordyceps are snake-like fungus or snake-like snake-like cordyceps fungi. Sorry. So you can see already a physical similarity between these two. And then when you compare them genetically, you can see the lineage that is associated between them. So we think originally there was a host shift that led to this new host of ants being infected. And here's a phylogeny of that occurring, starting with beetle larvae and then winding up in hymenopteran hosts like wasps and also ants. And you might ask yourself, like, how does that happen? And in fact, this type of host shift and host specificity is fairly common among different types of pathogens. And that's has to do with the fact that when a pathogen infects a host, that host basically becomes its new environment. So if you think back to your general biology principles, evolution involves selective pressures from the environment and selects for particular organisms well adapted to deal with those environments. So in the case where you're a pathogen infecting a human or any other type of host, that host basically becomes your environment. So changes in your host subsequently cause changes or in selective pressures that lead to evolution as well. So in the event that your hosts have some sort of reproductive barrier or some type of geographical distance, over time, as the hosts change, you can see that the pathogens associated with those host change as well. And we call this coevolution because they're evolving together. Now, there's a couple different ways in which this might have happened in Ophiocordyceps, one of which could be allopatric speciation. So perhaps this original host shift infected the primitive version of the ants we have today. A geographical barrier separates them. Ants are pretty small creatures. So, if you travel not too far, then you can really form some geographical distance. And as those two species of ants start to differentiate from one another and become different species, the fungal pathogens, such as perhaps the Ophiocordyceps in this case, can also differentiate and become new species. So this is just one potential way in which all of these different species of Ophiocordyceps could have emerged. Also, repeated host shifting is another possibility. But if you're interested in these kind of topics, I have a small TikTok series that you can watch that covers some of these basic biology principles using fun pop culture references like Pokemon, for example. And we're actually going to talk a little bit more about Pokemon later on in the talk. But first, I want to talk to you about what is the value of studying zombie ants, aside from just being a wickedly cool, fascinating thing to study. What is the why should we care, right? And to answer that question, we have to talk a little bit about fungi in general. So fungi have been used for human purposes for hundreds of, if not thousands of years in processes like fermentation, also bread making from the carbon dioxide that's released by yeast, and then also preserving foods. And while the fermentation and preserving food processes aren't as important today as they used to be prior to things like refrigeration and antibiotics that can help cure you if you get a bacterial disease, they are still pretty widely used today. And not just that, but in the new age of science, we've started to find that a lot of fungi have these amazing secondary metabolites that have pharmaceutical potential that can be used to help us in other ways. The most famous of examples is Alexander Fleming. So for those of you who are not familiar with this man's research, basically he was a bacteriologist. He observed one day that a fungal spore that contaminated one of his plates exhibited what's called a zone of inhibition. So basically around this fungal colony, there were no bacteria growing. And he was wise enough to realize that basically the fungus must be making some kind of chemical that is killing or preventing the growth of bacteria. And this is where we got antibiotics. A huge leap forward in medicine. It solved so many problems, things like tuberculosis or festering wounds that would have killed people less than 100 years ago now have potential cures. But it doesn't just stop there. We use fungal secondary metabolites for all kinds of medications, from things like antifungals, blood pressure medicines, immune system repressors for things like organ transplants. So studying fungi in general is really valuable due to the amazing diversity of secondary metabolites in which they create. Now, if we loop that back to Ophiocordyceps, we here have a system where we know there's a fungus making chemical compounds that affect animal behavior. So the question is, a really exciting question, what types of potential pharmaceuticals may be being produced by Ophiocordyceps that might be useful for things like behavioral disorders, which to date have relatively, or at least comparatively, very few options for treatment. So this is what we do in our lab. So I and Will, who is now a postdoc in our lab, did this big metagenomic study and looked at all the different all the different proteins, what we would call the proteome of Ophiocordyceps campanota floridani. And from that, he whittled it down to 586 secreted proteins, which are most likely to be involved in manipulation because they have to be injected into the host, and also 25 secondary metabolite clusters. So these are sets of genes that produce enzymes that are involved in turning one chemical into another chemical to produce secondary metabolites that way. And from this, we've developed a short list of potential compounds that probably have some kind of neurological effect, from things that bind to calcium ions that play a role in neuron firing, to things like toxins, and also compounds known to have neurological effects, one of which is called aflatrim. And we know aflatrim has neurological effects from previous study in another fungus, a plant pathogen that infects grain called Aspergillus flavus. And Aspergillus flavus also contains a similar secondary metabolite cluster that produces aflatrim. And when grain feed that is used for livestock is contaminated with this fungus, you see that the cows that ingest this fungus start to have neurological disorders. And when we compare the genomes of Aspergillus flavus to Ophiocordyceps, we see that the same components in the pathways are preserved in both species of fungi. Now, the symptoms that you see in cows can include muscle tremors, things like seizures and incoordination and trouble with muscle movement, which is exactly how you might define the drunkard's walk that we talked about earlier. So particularly the incoordination and the ataxia, the inability to walk properly, made us curious if these aflatrim compounds may be playing a role in the behavioral manipulation of ants. So to test this, we went to the University of Central Florida, and we collected 3 colonies of ants, and we injected them with various concentrations of aflatrim suspended in a PBS acetone solution. We then did behavioral observations to see how their behavior changed after injection with aflatrim, and compared these to ants injected with what we call a sham. So it's the exact same media, but without any aflatrim. And we used a combination of computational computer science and manual observation to try to figure out if aflatrim causes these types of drunkard walk activities in ants. And here's what we found. So here's a list of the 8 different behavioral manipulations that we saw in the ants. The first of which is resting behavior. So this is pretty common. Ants don't necessarily sleep like animals do, where they lay down. They kind of just sit there and very slowly move back and forth. We also saw ants climbing the walls frequently and also on the lids. So ants are very non-beholden to gravity, being as lightweight as they are and having the ability to really get those tiny little claws or fingers, if you want to call them, into even the tiny gaps in pieces of plastic. We also saw more dexterous climbing behavior. So we put a piece of twine in our arenas to see if the ants would perhaps bite onto the twine or if they are able to climb it. And as you can see here, that girl did a great job. Grooming behavior. So just like your dog licks their paws and rubs it on their ears, ants do the same thing. They lick their paws. They actually produce formic acid that they can use to bathe themselves, and they'll wipe it on their antenna and their body. And we called this grooming. We also saw biting behavior. This is one thing we were interested in, particularly because of the death grip phenotype. And then the 2 important ones, we saw normal walking. So this is what normal walking looks like in ants. You can see it's pretty much straightforward, like a train. It's always following a particular path. And this is what ants look like when you inject them with aflatrim. So you can see that those straightforward paths are no longer taken, and instead there's a lot of lateral movement side to side. And it's very reminiscent of the drunkard's walk that we see during late stage infection. So after we discovered that, it looks like aflatrim compounds could be playing a role in behavioral manipulation, we asked ourselves, well, what's the biological advantage of that? Because if you remember, I mentioned behavioral manipulation is all about reproduction and all about spreading the pathogen easier. Well, one thing we noted that this secondary metabolite cluster is highly upregulated during the end of infection. So it wouldn't be expressed during the previous behavioral manipulations, such as the antisocial behavior or the summit disease, but rather only towards the end when the death grip is occurring. And it's during this time that reduced activity may actually be important to maintain the ant in microclimates that are optimal for the growth of the fungus. It also might play a role in causing the ant to perform this hugging behavior, which is not something that we see in all behavioral manipulating pathogens. For example, this is the Arrhenius lampirodarum, which is the Arenopsis lampirodarum, which is the zombie soldier beetle. And you'll see here that even though it does the death grip, just like the ants do, it does not wrap its legs around the plant. And to our knowledge, also does not do the drunkard walk phenotype. So perhaps this in coordination helps to grab around the branches, which would actually aid in making sure that the ant is adhered to the surface before growth of your fruiting body. Because if there's one thing you don't want to happen after spending all of this time getting your ant to the perfect microclimate for growth is the ant falling out of the tree. And this sometimes happens. So you can see here, this is a really rare picture that was taken from iNaturalist. You can see that the ant actually has been separated from the body, but because the body is also hugging the branch, the fungus was actually able to maintain connection and continue its growth phase. Whereas in a normal situation without this hugging behavior, the major portion of the biomass that is needed for production of the fruiting body would have likely fallen out of the tree. Okay, so this is kind of where you come in. So just like us, we go out and we look for zombie ants in subtropical areas, and we note things like how high they were, what the temperature was that day, what was the humidity level, and we log all of this information to help us understand better the natural environment of these zombie fungi. And you can participate in this as well using an app called iNaturalist. So if you're not familiar with iNaturalist, iNaturalist is basically like Pokemon Go for real life. You can go out, take the app and take a picture of a frog, for example, and the app will say, I think that this is a Cuban tree frog. And then you can log that data. You can put in any kind of other values for things like humidity that you want, and it'll actually store it on a cloud that can be used by scientists to gather lots of data quickly. So the way in which this app works is you download it, you open the app, and you'll see right away that it actually keeps a portfolio of all the things that you've found. So, if you want to brag to your friends about a really rare butterfly you found, you can show them the picture that you took and show them where you found it. So it's really nice kind of game to play. And then you can select to take a picture. And once you take a picture, it'll actually give you this option about what did you see, and it will give you a recommendation. So in this case, it thinks that this flower I took a picture of is a tick seed. It's then up to you to curate. So it'll give you other suggestions, and you have to pick the one that makes the most sense based on visual cues. So it's not entirely done by computers. It also requires a little bit of input from the user. And then it gives you the option to add notes and then also upload your GPS coordinates. However, if you're interested in using the app simply just for enjoying nature, which I would highly recommend, and you're not interested in attaching your GPS coordinates, you can actually click that geo-privacy button there and turn that off. So you don't necessarily have to pinpoint exactly where you found something. For example, maybe you found something really cool in your backyard. You don't want scientists like me climbing over your fence to come take a close look at it. You can turn that geo-privacy off. And I think this is a really fantastic tool for you guys in particular, because the University of Arkansas State in Mexico is surrounded by zombie insects. So you guys have the perfect climate for lots of different biodiversity, especially for fungus, where it's warm and humid all the time. So here's two examples of some of the zombie fungi that have been found around. This is a Ophiocordyceps species infecting a wasp. And then here's Ophiocordyceps diptera infecting a fly, a horsefly. And all these little red buttons here are where people have found Ophiocordyceps species before. So another cool thing about the app is you can open our project page, which is called the Zombie Fungus Foray, or if you just look up Ophiocordyceps, it'll take you there as well. And you can actually see where other people have found Ophiocordyceps before and logged the GPS coordinates. So it gives you a good starting point of where to go and look. With that being said, I would also mention that this is nowhere near a complete map. So you're very likely to find these things even where people have not previously logged observations. So with that, I want to end a little bit early to give us just a little time for question and answers, but I'd like to thank my lab, particularly Dr. DeBecker, who is really the progenitor of this whole research project, and our undergraduate research staff, who has really helped with a lot of the data and been an integral part of our science. So with that, thank you very much for having me. I hope you enjoyed it and learned a little bit about zombie ants, and I'd be super happy to answer any questions that you guys have. Thank you.
Okay, so we're going to open the question session, guys. Do you have any question? And please, when you ask, try to lower your volume so Dr. Beckerson can hear you. Any questions, guys? Questions? No? Well, I do have one. Oh, sorry, over there. I don't know if you can hear the study, Dr. Beckerson.
I'm sorry, no, it's really hard to hear. If you can type it in the chat or just repeat the question, I can answer.
Yes, the question is why, if you know why the study kind of like right from the head of the ant. So why not other part of the anatomy of the ant?
So you're asking why does it grow out of the head and not elsewhere?
Yes, I think that's the question.
Great question. So if you think of ants, they have a external carapace of chitin. So they're basically like suits of armor filled with like jelly. So that's what an insect is. So the reason that it comes out of the back of the head is because there is a gap there between their like plates of armor, if you want to think of it, that is really soft. So you'll see that most of the time, that's the path of least resistance where these fungi come out. That being said, there are other species of Ophiocordyceps, like Ophiocordyceps humberti, which infects wasps, that comes out of all of those joints. So it's not just the back of the head, it'll come out of the leg joints and underneath the thorax and in between the abdomen and the pedial. So it's not, even though the ones that infect carpenter ants are very typically coming out from that one spot, there's a pretty large diversity in terms of exit points for Ophiocordyceps species as a genus.
Thank you. More questions? Yes.
My question is if it could potentially be used as a biological capacity.
The question is if these fungi could eventually be used to pest control, for instance, perhaps in the crops or something like that.
Great idea. There are actually biocontrol agents. I mentioned one earlier, Bavaria bassiana, which is a generalist pathogen, and it is a particularly good biocontrol because it affects everything and it infects things very quickly. The time of infection to death is like 3 days for that. It's called the icing fungus because it literally just looks like white mold growing on top of the ant, and it's very aggressive. Now, when we talk about very host-specific things like Ophiocordyceps, which has evolved to really have a very fine-tuned infection process of manipulation, and I didn't mention it in the talk, but that process can take 2 to 3 weeks. It is not as good of a biocontrol agent, specifically because of how slow it infects. But I think your question is a wonderful way to think about fungal pathogens, and in fact is a way that many scientists have approached trying to protect crops from insects, and then also like pest control. So yeah, I think general fungi are particularly well suited for that kind of biocontrol.
More questions? Yes. Yeah.
It's like less formal, but like what does it think about The Last of Us, like the series?
Okay,
What do I think about it? So
Yeah.
I particularly love The Last of Us because the people who made the game and the people who produced the show did their homework, and they collaborated with real scientists who understand the biology. Now, if I had to be nitpicky, there's kind of two things that kind of hang me up. One is the fact that the spores are not as contagious as you would think. So like really everybody should be a zombie at this point, because if you inhale spores, you're infected. And then two, the way that they show the fungus is very almost animal-like, where it's these tendrils that are moving around, and fungi don't work that way. They're very slow. They don't interact like that. They do things very slowly through chemicals. But other than that, I really love the biological relevance and thought that went into making this show at least somewhat plausible. So yeah, I'm very pleased with the show so far.
Further questions, guys?
Yes. The future, the host may change and those mammals, for example.
Could you repeat that?
Yeah, the question is if it's, so let me check if I understood the question. So the question is if in the future the host changes, it could be potentially affect mammals?
Right. So the premise of The Last of Us, basically. So you have nothing to worry about with zombie ants. We go out and we collect these things with our bare hands, and we have no concern about them being potential harm to us. And it has to do specifically with the intense host specificity. So these fungi won't even infect other species of ants. And you think about how different two species of ants are, and then compare that to how different an ant is from something like a vertebrate that has, a much more complicated immune system and complicated behaviors. So it's not really a feasible thing. But that being said, increase in global temperature will cause an increase in fungal pathogens in general. So it is worth paying attention to for things like Candida and Aspergillus that can infect immunocompromised people, because as the show accurately pointed out, as ambient temperatures become warmer, you slowly select for fungus with higher heat tolerance, and that allows them to overcome the barrier that currently exists between fungi that infect plants and invertebrates that have a body temperature of around like 28 degrees versus mammals that have body temperatures of 37 degrees. So it's definitely an important concept to pay attention to. But to answer your question, strictly speaking about Ophiocordyceps, it would take millions of years of evolution for that type of fungus to have any kind of behavioral manipulation effects in humans.
Thank you. More questions? Yes?
Once the ant is infected,
Sorry?
once the ant is infected, is there any kind of cure, or it's just like a dead cell?
Yes,
Yeah.
the question.
I think I heard you. So when the ant becomes infected, it's basically doomed. Particularly because the ant is either going to die or another ant is going to sense that it's sick and kill them. So it's a really rudimentary approach to herd immunity, but it's really effective in ants. So yeah, there's not like little ant medicine that's going to save these little girls once they're.
Questions, guys?
So the fact that it doesn't affect the nervous system, it means that the host is conscious about everything it's doing, but not being able to control it.
Okay, well, the question was since the infection affects, since the fungi just affects the tissues and not the nervous system. The question is if the host is kind of like conscious about what's going on, it's alive, but won't be able to control what is happening.
Yeah, so I think as biologists, we have a really bad habit of anthropomorphizing things. So like thinking about things in the ways that like vertebrates or higher consciousness beings might think about things. To what extent ants experience awareness, I have no idea. But to just basically answer your question, the ant is alive during this entire process, even up past the death grip. And so I'm sure it's a very confusing process. And when you boil it down to what's actually happening, I'm sure what's occurring is just the secretion of compounds that just heighten basically pre-existing behavior. So like something like a stimulant that makes them super hyperactive could very well be causing these abandoning social role phenotypes that we see. So it's probably less sophisticated in the terms of like, it's not a puppeteer, pulling the strings and making it do specific actions like somebody controlling a robot. It's more or less probably just, a fungus that's secreting like cocaine and the thing is going crazy and that yields these kind of phenotypes.
Any other question, guys? Yes?
It is possible that at a point that community to get to be one, like one organism itself.
I'm sorry.
Yes, the question is if it's possible for the fungus to be an independent organism, like live on itself without affecting perhaps any other organism?
That's an excellent question, because you see that a lot in like fungal pathogens of plants, for example, where they can have a host that they complete their life cycle in, but then also overwinter or infect other things where maybe they're just producing like asexually as like budding, but not really doing the life cycle. But with Oviocordyceps, I don't think that is the case. I think they rely entirely on their hosts. And when they are not infecting their host, they're kind of laying dormant in their really protective spore form until a new host comes around. I will say that we have been able to grow them on media in the lab, though, which is really useful for some of the projects we have coming up where we want to genetically modify them with things like CRISPR to knock out certain genes and see if that eliminates behavioral phenotypes, which is a pretty classical approach for studying how genes work in an organism. So you can grow it without the ant, but it requires a lot of babying. It is really like Goldilocks. It has to have everything perfect. It's very picky.
So, any other question? Yep.
Yeah, just a quick question. So you mentioned that this fungus is like being considered to to do improvements in like behaviors, right? So like are there like any behaviors that biologists are already considering like to apply this fungus like human behaviors, like I don't know, like diseases or something like that?
Yeah, so I think really where the value of understanding these chemicals comes from is kind of twofold. It's like two sides of the same coin. One is that by studying how behavior is manipulated and learning about these chemical pathways, we better understand how the nervous system works. Even if you're doing something as rudimentary as an ant nervous system compared to something as complex as a vertebrate nervous system, you can still learn basic biology principles that way. And then in terms of like the behaviors that humans have, it really depends. Like, perhaps we can find some stimulants that help with depression. If it is the case that, these fungi are really secreting compounds that really just increase hyperactivity in the ants. Perhaps we can find medications for ADHD it's really kind of a black box, and we're approaching it from a very open perspective. And those kinds of studies will come later on once we understand which chemicals are causing what behaviors in the ants. But that's ultimately the end goal here, is to try to find something that's helpful for people in general.
So, well, let us thank again Dr. Beckerson. Thank you so much for your interesting presentation. I am a theoretical physicist, and I think that I have learned a little bit about fungi and this parasitic behavior. Thank you so much. Thank you, guys.
Thank you. Yeah, it was such a pleasure. Thank you, guys.
Yeah. Yes,